Hi, my name is Alyssa Hallam, and I'm a professor in the MIMG department. In this two-part video series, we're going to talk about how model organisms are used for genetics research. In the first video, we'll discuss why model organisms are used for genetics research, and also how one selects an appropriate model organism to use. And then in the second video, we'll go over an in-depth example having to do with eye disorders that will illustrate what we can learn from studying model organisms. By the end of this two-part video sequence, you should be able to explain why model organisms are used for genetics research, explain how model organisms are selected for genetics research. You should also be able to design transgenic and knockout strategies for studying gene function in model organisms. And finally, you should be able to explain the conserved role of the PAX6 gene in regulating eye development across species, and how PAX6 function in Drosophila was studied using knockouts and transgenics. That's going to be the topic of the second video. Let's begin by discussing the different model organisms that are commonly used for genetics research. These include single-celled organisms like bacteria and yeast, also multicellular organisms like the roundworm C. elegans, the fruit fly Drosophila, and the mustard plant Arabidopsis. And then finally, vertebrate model systems such as the zebrafish, the mouse, and the rat. So first, let's discuss why model organisms are used for genetics research. The main reason for using a model organism is that it allows you to study gene function in ways that can't be done in humans. The reason this works is because most genes are conserved across species. Since human disease genes are also found in other organisms, we can learn about these genes by manipulating them in model organisms and then seeing what happens. Genes of interest can be manipulated in model organisms using genetic engineering techniques, and this can allow us to better understand gene function. The two genetic engineering techniques that we're going to focus on today are the generation of knockout animals and the generation of transgenic animals. So what are transgenics and knockouts? A knockout animal is an animal in which a gene of interest has been inactivated so that its function can be studied. In contrast, a transgenic animal is an animal containing foreign DNA in its genome. This DNA can be in the form of an insertion, where you have extra DNA that's been added to the genome, or it can be in the form of a substitution, where some of the organism's DNA has been replaced with foreign DNA. One thing I want to point out is that the term transgenic is a very broad term. So you should be aware that there are many different types of transgenic animals. And what a transgenic is used for really depends on what type of foreign DNA has been introduced into its genome. Now let's take a closer look at knockouts and transgenics. So here I'm showing you a part of the genome of a model organism that contains a single gene, which we're calling gene X. An animal containing a knockout of gene X would be one in which the DNA encoding the gene has been deleted from the animal's genome. In contrast, a transgenic animal is any animal in which a piece of foreign DNA has been inserted into its genome. So in this case, gene X is intact, but the animal has foreign DNA inserted elsewhere in its genome. In this case, the foreign DNA is inserted close to gene X, but that is not necessarily the case. The extra DNA could actually be inserted anywhere in the genome. And then finally, an animal can be both a transgenic and a knockout. For example, here you can see that the foreign DNA has been inserted into the genome in place of gene X. So the animal is a knockout because it lacks gene X, and the animal is also a transgenic because it has foreign DNA inserted into its genome in place of gene X. So just keep in mind that the terms transgenic and knockout are not mutually exclusive, and many genetically engineered animals are actually both transgenics and knockouts. Now let's focus for a minute on knockouts. There are two main approaches to generating knockouts, and these are called forward and reverse genetic approaches. In the reverse genetics approach, you take a known gene of interest, and then you generate a model organism in which the gene has been knocked out. And these days, the knockout is typically made using CRISPR technology. And then you look at the phenotype that results from loss of the gene. In contrast, in the forward genetics approach, you start with a particular phenotype of interest. And then you screen a collection of mutants to see if any of them show the phenotype of interest. Or you might generate new mutants by chemical mutagenesis, where you mutagenize a population of organisms and then screen for ones that have the phenotype you're interested in. And then you identify the gene or genes that are causing the phenotype of interest. So as an example, if you're studying the visual system, you might mutagenize a population of flies, screen the mutagenized population for vision defects. And then when you have flies with vision defects, you would try to map the genes that are responsible for those vision defects. Now let's talk about how one goes about selecting a model organism to use. Your choice of a model organism depends a lot on the particular biological question that you're studying. Almost all biological questions can be studied using multiple model organisms. But one common approach is to select the simplest model organism that it's possible for you to use. So for example, if you want to study fundamental processes in cell biology, like for example transcription, translation, or the cell cycle, then you might decide to study these questions in a relatively simple single-celled organism like bacteria or yeast. If you're studying a topic in developmental genetics, for example embryonic development or organ formation, then in this case you're going to need to study a multicellular organism. And you might choose to study Arabidopsis, C. elegans, Drosophila, or zebrafish. Working with these organisms is going to be faster and easier than working with a mammalian model system. And in addition, both C. elegans and zebrafish larvae are transparent, which can be an advantage because it's very easy to see cells dividing or migrating during development in these organisms. If you're studying the genetic basis of behavior, then obviously you're not going to be able to work with bacteria, yeast, or plants. So in this case, it might make sense to work with a less complex invertebrate model system like C. elegans or Drosophila. Or it might make sense to work with a zebrafish because it's a vertebrate, so it's closer to humans and it shows a lot of complex behaviors. Or you might choose to work with a mouse, which is a mammalian system and therefore is closer to humans than the other model systems. And then finally, if you're studying very complex behaviors or diseases, you might need to use a mammalian model system. For example, if you're interested in studying maternal behavior, you would need to use a mouse or a rat since the other species don't show maternal behavior. Or if you're studying complex diseases like cancer or autism, you would probably want to use a mouse because there are well-established mouse models for these diseases. And then finally, you might choose to use a rat instead of a mouse for studying some behaviors, since rats are generally smarter than mice and often they show more robust behaviors. So as you can see, there are many different factors that you need to consider when choosing a model organism to study. To conclude, in this video, we've talked about why model organisms are used for genetics research and also how a model organism is selected. And now in the next video, we'll take a more in-depth look at how model organisms have been used to study human diseases.